Hey guys, so in this video I'm going to be talking about how fructose 6-phosphate is regulated during the glycolysis cycle. Alright, so let's get started. So first we have fructose 6-phosphate, and during glycolysis it's going to go to fructose 1,6-bis phosphate. And glycolysis. And then during gluconeogenesis, it's going to go back to fructose 6-phosphate. Alright, so what are the enzymes that actually do this? So to get it from fructose 6-phosphate to fructose 1,6-bisphosphate, we have the enzyme phosphofructokinase. And this is going to take an ATP and then convert that to ADP. And then under gluconeogenesis, we're going to have fructose 1,6-bisphosphatase. So we have fructose 1,6-bisphosphatase. It's going to take an H2O and then kick out a phosphate group. So yeah, H2O to phosphate group, ATP to ADP. So, well, when we undergo glycolysis, we're going to have a lot of ATP, right? Because the products of glycolysis are going to go through the Krebs cycle and then through the electron transport chain. And then, here, put glycolysis so you don't get glycolysis. Very confused. And then under gluconeogenesis, we're going to have a lot of AMP. Because we're taking that phosphate group and we're making glucose out of it. So, what are some inhibitors and activators? Well, actually, let's go back for 1 second. So some inhibitors are going to be ATP, right? ATP because if we have a lot of ATP, we're not going to want to make more. Another inhibitor is going to be citrate. I'll put an X for inhibitor. Inhibitor. This is going to inhibit glycolysis. Citrate because that means that the Krebs cycle is backed up and we're making a lot of citrate. It's a product, and if we have a lot of that, it means like we need to stop making more. And what are some activators? Well, some activators, and put those in purple, are going to be AMP and ADP because that means that energy is low. And another activator is going to be this special product called fructose 2,6-bisphosphate. And we're going to talk about this in the next slide right now. So here we have, let's talk about fructose 2,6-bisphosphate. So what's the difference between fructose 2,6-bisphosphate and fructose 1,6? Well, the only difference is that the phosphate group is on the number 2 carbon in fructose 2,6-bisphosphate. It's on the number 2 and the number 6, and on the fructose 1,6-bisphosphate, the phosphate group is on the number 1. Well, how is this regulated? Why do we even want this? Well, so let's draw a little diagram here that kind of helps clear things up. So first of all we have fructose 6-phosphate, and then we have two options, right? We can either let's draw a little cycle. We can either get fructose 1,6-bisphosphate, or we have this other option of going to fructose 2,6-bisphosphate, right? So we already talked about this and how this is inhibited. So let's talk about inhibited and activated. So let's talk about how fructose 6-phosphate goes to fructose 2,6-bisphosphate. All right. So essentially, how we have PFK1 over here, and fructose bisphosphatase 1. Well, it's the same exact thing Similar, very similar, except that we're going to have a 2. So it's going to be phosphofructokinase 2 and fructose bisphosphatase 2. Um, these are going to be the enzymes that initiate fructose 6-phosphate to fructose 2,6-bisphosphate and fructose 2,6-bisphosphate back to fructose 6-phosphate. It's going to be these two enzymes. And now we're going to talk about how they are activated and inhibited and how they work. It's actually pretty simple. So uh, let's talk about these enzymes. All right. So we're gonna zoom in on these two enzymes on the next page. So we're gonna have phosphofructokinase and fructose bisphosphatase. These are both 2, right? And let's draw these twice so you can see the complete cycle of how they actually work. All right, and why they do that. So here we have this. So when this enzyme, this is all one enzyme. When this enzyme, these are all each one's enzyme. When this first one is not phosphorylated, meaning it has a hydroxyl group on it, the PFK section of it is going to be activated. Well, what does this PFK section do, and what does it do? This PFK2. Well, this PFK2 is going to allow us to get from fructose 2,6-bisphosphate fructose 6-phosphate to fructose 2,6-bisphosphate. That's what this PFK is going to do. So let's go back here. So it's going to stimulate glycolysis. Just like how if we have fructose 6-phosphate to fructose 1,6-bisphosphate, that's going to stimulate glycolysis. This is essentially the same thing. It's going to stimulate glycolysis if it's not phosphorylated. Okay, if it's just regular, it doesn't have a hydroxyl group. But if we were ever in a situation where, for example, glucagon was released, glucagon is released. Well, when glucagon is released, it means that glucose is gone, right? Glucose in the blood is low glucose. So when this happens, the CAMP, cyclic AMP, dependent protein kinase is going to be activated, and this is going to work by taking an ATP and converting it to ADP. It's stimulated by glucagon, and when this happens, fructose bisphosphatase 2 is going to be active. It's going to be active. So we can draw a little phosphate group right here. Okay, let's draw this phosphate group. So when this happens, it's going to be active, and this active form will inhibit glycolysis. Since this part's active, right? It's going to be the ACE. It's going to be fructose bisphosphatase, and to go back on that, when we have fructose bisphosphatase, we're going to move this fructose 2,6-bisphosphate back to fructose 6-phosphate. So back to the original molecule, and we're going to push it the other way to gluconeogenesis. We're going to push it upstream to gluconeogenesis. And that's why this makes sense, right? This enzyme right here is going to push the fructose 2,6-bisphosphate back to fructose 6-phosphate. And how it's going to do that is through this enzyme called phosphoprotein phosphatase. Phosphoprotein phosphatase. So insulin is gonna stimulate glycolysis. This is gonna stimulate glycolysis, meaning we have a lot of glucose in the blood. Like we just ate a meal, so our insulin just spiked. This enzyme is gonna hydrate fructose bisphosphate 2, and it's gonna kick out a phosphate group. And that's how this enzyme works.